Hi, everyone. Welcome to the Build a Cell seminar, and thanks so much, Rafa, for agreeing to give the seminar, and you can get started.
Thank you, Kate. Thank you for the invitation. Thank you. Thank you all for joining this amazing series and my talk, which is about lipid flip-flop and growing of synthetic cells. So to give you a very short introduction of who I am and where I come from the city, the coastal line of Brazil called Recife. This is where I did my studies, bachelor's in biomedical sciences and master's in biology biophysics. Then I moved to São Paulo to do a PhD under the supervision of Karen Risker, where I studied various aspects of membrane biophysics, most specifically on drug delivery-based processes such as electroporation and membrane fusion. Then I moved to Germany to a place called Potsdam near Berlin, where I did a postdoc under the supervision of Rumi Dimova, a famous person in the field. I guess many of you here would know her. And after that, I've been in many places. And after that, I moved to Groningen, where I held a teaching research position at the University of Groningen under the supervision of Walter Roos. And more recently means 3 years ago, I went to the west of the Netherlands, where I am based now, where I did research under the supervision of Kees Dekker, who was also speaker here 2 weeks ago, if I'm not mistaken. And it was also in Rumi's lab several years ago where I got in touch with the field of synthetic biology, and it's fascinating. I don't need to sell that to this audience. And given my background and the title of the talk, this is not the kind of flip-flop I'll be talking about. Not is it going to be about rafts, even though rafts are or used to be very popular in the field of membrane biophysics in general. It's a different type of flip-flop I'll be talking about today. So in the context of synthetic biology, we take inspiration from living cells or life. And to be considered alive, a few criteria must be met. And this is more or less what we draw the line of what is synthetic and what is natural, and if synthetic systems will ever be alive. So they first need to be compartmentalized. And by compartmentalization, we can talk about coacervates, vesicles made of polymers, and vesicles made of lipids. The latter is the compartment that cells are made of. They should also exhibit some type of metabolism, energy producing and consumption, and they should also reproduce or replicate. Through these years in research, I've been involved in Europe's 2 major synthetic biology consortium. In Germany, I was involved in the MaximBio, which is a consortium in synthetic biology, which is basically a collaboration across several different Max Planck institutes. And later on, more recently, in the BASIC, build a synthetic cell whose goal is very similar to BASIC, is very similar to MaximBio. Basically, the idea is that we want to create a lifelike system that is able to autonomously grow, transmit information, and divide. So this is typically divided in a few different working packages. The first one is related to fueling or producing, consuming energy. The second one is about DNA processing, replication, translation, protein production as well, and cell division. And this is the topic of my talk today. We are in general very familiar with the process of cell division, where we see these striking morphological changes that cells undergo. So there is, at the cell level, a large familiarity with the division process. However, comparatively, a lot less is known about the equally important process of growth. And growth is as important as division, because divided cells will have half of the content of the cells just prior division. So to take inspiration from cells, we need to learn how cells duplicate their membrane compartments. Membranes will be the main topic of the talk today here. So lipid synthesis takes place at this organelle inside the cell called the ER, the endoplasmic reticulum. So the enzyme lipid synthesized machinery is present in the outer leaflet of this organelle, so the cytosolic side, where the lipids are produced and inserted therein. But it also needs to move to the inner leaflet, so then it will accommodate the growth of the compartment. Then this organelle will grow and bud off in the form of many organelles, the so-called vesicles, and these vesicles will fuse with the plasma membrane further down. And as they fuse, the size of the plasma membrane will increase, and that's how growth takes place. Can we mimic this? The idea is that we would use a synthetic system that is much simpler compared to cell. And by synthetic system, I mean liposomes. So liposomes are basically a lipid aggregate where you put lipids in a water solution, they will self-aggregate in the form of lipid bilayers. In general, it depends on the lipid species. And in order to prevent exposure of the hydrophobic tails to water, the edges will close in the form of vesicles, entrapping the solution during the process, the aqueous solution during the process. Liposomes can come in many sizes and shapes. Here, I will specifically talk about liposomes that are comprised of a single bilayer, the so-called unilamellar vesicles. And they can be small or large, up to 200 nanometers. It varies a little bit depending on the author. And giant vesicles, and these are typically used as cell mimetic systems, and they are much larger and visible at the microscope. Although they are fundamentally the same, they are physically different because small vesicles are under certain constraints that larger the GUVs are not. And one of them is because they are so small, they can only accommodate so many lipids in the outer leaflet compared to the inner leaflet compared to the outer leaflet. And they intrinsically exhibit some sort of asymmetry, in this case, in terms of lipid number or area. So small liposomes have more lipids and larger areas in their outer leaflet compared to the inner leaflet. And why is this important? This is coming up very soon. So like cells, lipid vesicles can also attain specific shapes, and these shapes will depend on environmental factors. The first one is the cell shape is determined by only two parameters. The first of which is the so-called spontaneous curvature, or in this case is the area difference, which is a reflection of the spontaneous curvature. And we can change the spontaneous curvature by many means. In this case, is if we have lipids in one leaflet that are bulkier than lipids in the opposite leaflet. In this case, is the outer leaflet. And because the membrane is fluid, it will bend. And in this case, forming a bud. And we can calculate the spontaneous curvature simply due to from geometric factors. And the second parameter is the reduced volume, which is fundamentally how inflated the vesicle is. If you think of a GUV as a balloon, it's how inflated or deflated it is. So if the vesicle is inflated, it's always a sphere, regardless of the spontaneous curvature. But if you increase or decrease the area difference of the spontaneous curvature, then you attain different shapes. If the spontaneous curvature is positive, then the vesicle will bud outwards, and likewise negative, it will bud inwards. If we are talking about growing a compartment, if we have growth alone, then we are moving within this trajectory here. We decrease the reduced volume because we are incorporating membrane material in the vesicle. But you may also have a situation where we are changing both simultaneously. And in the case of growth coupled with division, one would ideally want such a system. So then when you have division will be symmetrical. Can we grow synthetic compartments? The answer is yes. There are several approaches by which this could be done. One of them is upon incorporation of soluble content, for example, fatty acids or detergent. Detergent is the case of this video here. So you have GUVs in a chamber where an electric field is applied. And then this electric field imposes tension in the membrane. So you can see that the area is growing upon the incorporation of the detergent. But unfortunately, what we have is there is so much growth that the vesicle attains before it solubilizes. This is what detergents do. In this case, you can grow up to 50% of the membrane area before the vesicle is fully solubilized. So incorporation of fatty acids or detergents is a fast growing process. However, it's also disruptive. And this is something we want to avoid. A second approach is upon synthesis of lipids inside the vesicle. This is more similar to how cells grow compartments. But this is also scientifically very challenging. It typically requires the incorporation of many lipid synthesis proteins. And for those who work with membrane reconstitution of proteins, you will know that this can be very challenging. In this specific example, PS is synthesized internally, and the authors added a fluorescent reporter. And then once PS is detected and the GUV would light up. And this can be seen here. In a few hours, you have PS detected by the reporter. However, when you measure the GUV size increment upon a few hours, we don't see a visible increase in area. So the process is very modest. So the question that we asked ourselves was, can we efficiently grow synthetic cells without disrupting them? Some years ago, we reported this paper where we produce cationic small vesicles made of distilled lipids, though TAP is positively charged. The small vesicles were labeled in red, and then we mixed them with GUVs labeled in green. The GUVs were negatively charged, and we hoped to see fusion. If we saw fusion, then the GUV area will increase, and this is more or less what we see here. So we see some diffraction limited spots, red liposomes here, and a GUV here, which is initially spherical. And as the LUV start fusing with the GUV becomes red, and strikingly, it also becomes floppy as it grows. However, something that is this was amazing. We loved what we saw, but something that we didn't know back in the days is why the GUV also would show these budding structures, and this floppy membrane will also become dense looking again as the GUV will become spherical. So we were able to have a system where the GUV will grow efficiently. Back in the days, we didn't know how efficiently that was, and we also couldn't explain the mechanical effects yet. So can we quantify how much area is grown via membrane fusion? This is something very recent that we published only last week. So we did something very similar to the previous slide. We had green GUVs, red label LUVs, but the LUVs were also loaded with a content cargo. So when there was fusion, there would be content mixing, and the cargo would transfer into the GUV lumen. And we fused these LUVs by micro-injecting them with the help of a micropipette in the vicinity of the GUVs. This is what we see here. The GUV becomes floppy as the area grows. It also becomes red. And most importantly, it accumulates the probe. But as you can see in the latest, we also see lots of buds. But as we can see in the latest stage, the probe disappears. And I can tell you in a couple of seconds that the probe disappearance is not due to photobleaching. Then we can use both probes from the LUVs and the GUVs form a fret pair, which we can use to measure the efficiency of fusion. So this is what we have. The micropipette fuses the LUVs here to the GUV. The GUV eventually buds. And we can use the readout of fret to measure the efficiency. Efficiency in our case is the number of LUVs that fuse with each GUV. So if we follow the red line here, we can see that we have a few tens of thousands of individual LUVs fusing to that one single GUV. This is remarkable. It's a very high number. If we calculate the same property using the content mixing instead of the lipid mixing, we have relatively similar numbers, also a couple of thousands LUVs fusing to a single GUV. So we have a remarkable membrane fusion system. Fusion is also a very fast process. We have a few thousands of LUVs fusing to that GUV every second, which is good. We have a system that works very efficiently, but also is very fast. Because we know the initial GUV area and also the area of the fusing LUVs, we know the number of LUVs that fuse to each GUV, we can then calculate the increasing area. And here is the area increase where 0 means no growth and 1 means 100% growth, so double the area. And to the best of our knowledge, this is the first system where we have fusion, where we have growth that is above 100% area increase, so double the area. But as I mentioned, we have the disappearance of the contact marker here. And if we follow the same video now on the phase contrast, we'll see something remarkable. So first we see membrane fluctuations because it's a result of area increase. Then we see budding formation, and the contrast eventually disappears. And this more or less coincides with the disappearance of the content marker here. The fact that the contrast disappears here is an indication that the solutions inside and outside mixed. And this is a reflection of the increase in permeability in the membrane. In other words, the membrane has pores. So we have a relatively disruptive process. And this is what we can see here. When we look at the GUV contrast, we see that it decreases over time. This is the black data here. So we see that the GUV can sustain a high amount of fusion, so the contrast is more or less stable before it stochastically becomes permeable. So pores that are formed in this membrane appear stochastically. So our conclusion is it is possible to duplicate the GUV area or to grow even more than 2 times. However, the process at this extent of fusion or this extent of growth induces extensive membrane remodeling, and membrane disruption, pore formation. Why do membranes bud and perforate? This is something that we didn't know back in the days, but now we do. So in our fusion growth system, we have a very clear sequence of events. We start with a GUV that is initially flat, so it basically has no spontaneous curvature. And then we fuse many small vesicles to this GUV, growing the GUV area. That's why the membrane becomes floppy. And eventually there's a budding transition. So the GUV will start budding. And the buds over time shrink, and this coincides with leakage. And this is because they fuse, although they're initially symmetrical, the GUVs fuse with tens of thousands of intrinsically asymmetric LUVs. So this asymmetry from the LUVs is then transferred to the GUVs. So as a result, the initially flat GUV membrane we had then becomes asymmetric because there's an excess of lipids in the outer leaf flat, and as a consequence, the GUV will bud. And the direction of the bud is outwards because the excess of lipids is in the outer leaf flat. We can calculate this. So from the buds, we see that there's spontaneous curvature change. We know the number of LUVs that fuse to the GUV. We knew that from the frat and the contact mixing experiments. And we can then calculate the ratio of lipids in the outer versus the inner leaf flat. And this is what we have. We have an increment of between 8 and 10% lipids in the outer leaf flat. And what we're seeing is above approximately 5% asymmetry, the GUVs will spontaneously rupture. So this is this is how we discovered that the shape transitions happen because of changes in spontaneous curvature, which are a result of asymmetry in lipid numbers across the membrane. The process is not specific to membrane fusion, so we perform molecular dynamic simulations of membrane patches that have increased asymmetry across the membrane. So we remove lipids from the inner leaf flat. So it's not based on membrane fusion, but simply adding a difference in lipid number across the membrane. And then the black data show that above a given threshold of asymmetry, we have pores formed in the membrane, and the area of the pore increases as the asymmetry increases. The blue data shows that the time it takes for these pores to form becomes shorter as the asymmetry increases. The final message here is these effects are very general. It's not specific to membrane fusion only. It would very likely happen to lipid synthesis too. And having an asymmetric number distribution of lipids across the membrane would lead to pore formation. So if we are growing a synthetic compartment and the membrane becomes disrupted in the process by forming pores, does that mean that it's the end of the line for growing synthetic cells? Not necessarily. If we look at the pore stability energy, so if we start with a flat membrane and we apply lateral tension, experimentally we can do that by adhering to a substrate or using micropipette aspiration or electroporation. Then the membrane will stretch to a point where you will form a pore, so it will break. This is the so-called lysis tension. And the lipids at the edge will bend in order to prevent exposure of their hydrophobic tails to water. This bending has an energy cost that is called the edge tension, which will try to close the formed pore in a lipid-only membrane. So the pore energy depends on the difference between the edge tension and the surface tension. And this also creates the so-called critical radius. And the critical radius is the radius size of the pore below which a pore is stable and it will close spontaneously, and above which it will induce indefinite growth and membrane collapse. The critical radius is a ratio of the edge over the surface tension. So for typical symmetrical GUVs, critical radius is very large, so you can porate a GUV forming very large pores. This is the case of GUV electroporation. So we create very large pores in the GUV, you can see, and the pores here will spontaneously reseal. So pore formation in the lipid-only membrane is a self-resealing process. However, if we decrease the edge tension, in this case is by adding membrane charges, then we also decrease the critical radius, and pore formation in this vesicle will lead to vesicle collapse. This is basically as popping a balloon. So if we create a pore in this vesicle, the balloon will break. There's no self-resealing process anymore. Edge tension is too low, and content is immediately released, escaped. So coming back to our asymmetry story, what are we changing when we're adding asymmetry to the membrane? Are we changing the tension? So we are imposing large tensions. Lysis tension is approximately in the order of a couple of millinewtons per meter. Or we are inducing collapse, or we are changing the edge tension during the process. And the GUVs will spontaneously collapse if the edge tension is below these values between 5 and 10 piconewtons. So we can calculate the tension. So trying to address this point first. If we know the bending rigidity of the membrane, which we do, and if we are able to calculate the spontaneous curvature, which we are from the bud dimensions. So from these growth trajectories, we see that buds are initially formed after a few tens of seconds, and they shrink because the spontaneous curvature increases, so more asymmetry is created. And this leads to an increase in spontaneous curvature that translates to membrane tension that has curvature origin and the so-called spontaneous tension. And as we can see, the spontaneous tension is in the order of a few tens of micronewton per meter. So it's much below this. So although fusion and asymmetries will induce tension in the membrane, it does not lead to lysis. It does not reach the lysis tension. Then it leads us to looking at the edge tension. So when we measure the edge tension in GUVs after incubation with the LUVs, we see that the edge tension is decreased. So this is an electroporation experiment of GUVs that have been incubated with LUVs. As we can see, we have large pore formation, and the pores that are formed will induce the GUV collapse, just like in conditions of very low edge tension. In conditions of very high asymmetries, we don't even need to electroporate the GUVs. Instead, they porate spontaneously because the edge tension is so low, the critical radius is so small that any defect formed in the membrane will induce to collapse. So the answer to this is lipid asymmetry mainly causes a decrease in edge tension, not in lysis tension. If we again simulate a membrane, and this was done by Marco van Tilburg from the group of Sivet Yamarink at the University of Groningen, this membrane patch is asymmetric. It contains more lipids in the outer than in the leaflet. We can see that over time, the total lipids in the upper and the inner leaflet remain constant. So there is no exchange of lipids. However, if we have a pore in the membrane, which is a situation that happens during growth, then lipids lining the pore edge are now able to access the opposite leaflet. And this is exactly what we see here for a four-component membrane, where 3 individual lipids of 3 chemical compositions are labeled here. This membrane is asymmetric. It has more lipids in the upper compared to the inner leaflet. We see that pore formation will allow lipids to access the other leaflet. In other words, there is flip-flop here. When the pore opens, lipids undergo spontaneous flip-flop. So this is spontaneously driven. And the process is clearly directional. It's from the overly populated to the underly populated leaflet. So if we open a pore in the membrane, that allows lipid to flip-flop. And if we simulate a pore in an asymmetric membrane, lipids spontaneously flip-flop, and the pores will also spontaneously close. So the situation that we have is if we have too high asymmetry, the membrane will spontaneously collapse. In the cell case, this is equivalent to cell death. However, in intermediate conditions, then pore formation will allow lipids to flip-flop and decrease the initial asymmetry imposed during the growth process. And as a consequence, because the asymmetry is the driving force for pore formation, and the asymmetry doesn't exist anymore, then the pores will eventually close. So in other words, what we have is a situation where pores formed in lipid-only membranes act as a catalyst for their own closure. So they work as lipid scramblazes. Can we prevent pore formation in the first place by systems design? So we start with a system that will grow, but it will not have such asymmetry increase. So the idea is that we would move the axis of lipids from the upper to the bottom leaflet. The way cells do that is two ways. The first way is several cellular membranes are rich in lipids that are capable of undergoing flip-flop. And this is the case of cholesterol. The other situation is several membranes, such as the ER membrane, is also rich in lipid flipping enzymes, such as lipid flipases and scramblazes. So if we take inspiration of the first method first, and in the later part of my presentation, the second method, the situation that we have is that we are growing a membrane that induces too high asymmetry in the process, and the GUV that we have contains too many small buds. So taking inspiration of the first approach used by cells, we prepare GUVs and LUVs that are then fusing, growing the GUV, but now they both contain a flip-flop capable molecule. So the situation that we expect is the GUVs, they are initially flat, will have an equal distribution of these flip-flop capable molecules in both leaflets. However, after fusion, upon an increasing area, because of higher number of lipids in the outer leaflet, these molecules will migrate spontaneously to the bottom leaflet, alleviating such asymmetries. For this, we use a small molecule that, like cholesterol, is presumably able to undergo lipid flip-flop. This molecule is called seromycin-6. And we perform molecular dynamic simulations of a membrane patch containing 10% of these molecules, one of which is labeled here. And you can see that even without pore formation in the membrane, seromycin-6 is able to undergo spontaneous flip-flop. The process is also very fast. It's comparable with flip-flop timescales of cholesterol, and there is no need for pore formation in the process. So when we do experiments with GUVs in these conditions, so this is a control experiment, no flip-flop in this case, the GUV will grow because it fuses with many LUVs. You see membrane becomes floppy because of area increase, decrease in reduced volume, and you also see bud information. I'll play it again. We're actually very lucky to see a bud being formed at the equatorial plane of this confocal slice here. So we see bud information as the GUV grows because of induction of spontaneous curvature, and multiple buds are formed, even buds attached to other buds. And if we look closely, these buds also shrink in the process, and this makes total sense because the buds form because of spontaneous curvature, and as more LUVs fuse, the spontaneous curvature increases, and the bud will become more and more curved, meaning that they become smaller. So when we track the trajectory of this GUV in the shape diagram, where we have the reduced area difference, which is fundamentally a reduced measure of the mismatch in the area of the outer and the inner leaflet as a function of the reduced volume. The reduced volume tells you how much deflation comes from growth. We have this trajectory. Each color is a time point here, and the trajectory goes in this direction. For a GUV without seromycin-6, you end up with a large GUV with many small buds. And if we are to divide this GUV, for example, using photoactivation, I'm skipping the methods on how this is done. Division results in a large GUV forming many small, shedding many small buds. So it's a highly asymmetric process. When we do the same experiments, but now with GUVs and LUVs containing 10% of seromycin-6, we have much wider buds formed. You're probably thinking, okay, this vesicle is still not symmetrical. No, it is not. But if we look at the trajectory, we see that for relatively comparable values of reduced volumes, meaning how much the vesicle grew, we have a much milder increase in the area difference. This is because the number of lipids in both leaflets is more balanced. And if we are to divide these vesicles, then we have more symmetrical division. So we are closer to dividing two vesicles of equal sizes. I'm not going to talk much about division in this talk. For that, I refer you to this paper that we published a month ago or so. But I'm still going to talk about growth. Okay, going back to point 2 of the mechanisms used by cells to alleviate such asymmetries. Cells do not rely on pore formation of flip-flop capable molecules to alleviate the asymmetries that grow during growth. Instead, they use lipid flipping enzymes, some of which are lipid scramblases. These are proteins that, like ion channels, they are lipid channels. They enable the exchange of lipids bidirectionally in an energy independent manner, and the process is also fast. A few years ago, there was this dynamic simulation on a piece of membrane reconstituted with a lipid scramblase, and you can see that lipids can spontaneously access the opposite leaf flap through the protein. And if in our case of a highly asymmetric membrane, of a membrane that has more lipids in one of the leaflets, if we have such a scramblase, that will allow lipids to mix from both leaflets. So the outer leaflet in this case is labeled in red, and then you can see that in the inner leaflet you have an increase in red labeled lipids. Eventually, these buds, because of asymmetries, will flatten until the membrane becomes completely flat. So we took inspiration from this. This has never been shown experimentally to the best of our knowledge. So we, again, did a real-time experiment on GUV that grows without any flip-flop process. So here, fusion is detected as an increase in the GUV signal. And then we can see that there's more fusion. The GUV grows by forming lots of buds in their membranes. They form many buds, as we know now, because of the increase in spontaneous curvature and symmetry in the GUV membrane. If we do the same experiment, but now in GUVs that contain a lipid flipase or lipid scramblase, then the results are very different. So you see that the GUV will also fuse, so the signal increases over time. But rather than forming many small buds, the GUV becomes very floppy. So it's a sign that the asymmetry is not so high. And it will also project many buds. And these buds are much wider. So that's a direct morphological indication that the asymmetry here is much smaller compared to the situation where we don't have a lipid scramblase. So this can be clearly seen in the snapshots here, where we grow a GUV without lipid scramblase forming many small buds, or when we have a lipid scramblase, we have a floppy GUV rather than a tense-looking GUV having much wider buds. When we follow the bud sizes for the situation without the protein, the bud sizes are more or less constant. However, when we follow the trajectory of, when we measure the buds over time for the GUVs containing the scramblase, then we see that the buds widen over time. And this is because the asymmetry is decreasing due to lipid flip-flop. So having a protein that scrambles lipids will reduce or alleviate the asymmetries that build in the growth process. We can calculate the flip-flop rate if we know the growth rate, which we do from the experiment, and also if we know the fraction of alleviation of asymmetry, which we also access from the bud diameter change. And for this protein specifically, we have a lipid flip-flop rate of approximately 10 to the power 5 lipids per second. To give you a reference, typical scramblases measured in cells or in reconstituted systems have a lipid flip-flop rate of 10 to the power 3 and 10 to the power 5 lipids per second. So we are working with one of the fastest scramblases ever reported, which is paramount for what we want to do with synthetic cells. To put that in numbers, for a small compartment, much smaller than GUVs, for example, for a bacterium-sized compartment of 1 micrometer diameter and 5% asymmetry, which is what we attain in these experiments, then this means that we have 10 to the power 5 lipids excess in one leaflet. For GUVs, the same amount of asymmetry generates 10 to the power of 8 lipid excess. And we are not able to alleviate that in seconds. So we have a competition of growth and asymmetry build up and asymmetry relief, where in our experiments, the build up still wins. And that tells us that lipid scramblases are designed to balance asymmetries in small compartments such as in bacteria and in the ER. So if we want to work with synthetic cells, we have to either increase the amount of such proteins in the GUVs or work with more efficient scramblers. So to finalize, we have shown that it is possible to extensively grow synthetic cells at unprecedented rates, bypassing the two-area increase within only a few minutes. However, because of the magnitude of growth that we have in the system, we also introduce asymmetry, and this asymmetry will change the spontaneous curvature of the membrane, leading to budding formation. We will induce membrane tension, and more importantly, it will decrease the edge tension, leading to membrane perforation, and in extreme cases, a whole vesicle collapse, which is something we want to prevent. And what we have also shown is that flip-flop universally alleviates asymmetry in growing synthetic cells. It doesn't really matter if we have flip-flop via leakage pores, or if we have a flippable molecule, or if you have a scramblase in the membrane. So the final message I hope to have conveyed to you is that growing synthetic cells will need a mechanism to mitigate the mechanical effects that arise during growth of synthetic cells. I would like to thank everyone involved in this project throughout the years, and of course you for the attention, and I'm open to questions you may have. Thank you.
Thank you. Thank you so much. There are questions in chat. Can you see chat?
I am trying to find it out. Yes. Okay.
If you could read the question before answering out loud, because people watching the recording don't see the chat.
Yes, I will read them. Oh yeah. So Lindsay asks, when you work on fusion of GUVs and LUVs, do the vesicles have channels in the membrane to equilibrate osmotically? That's an interesting technical point. We work with osmotically based solutions. So the GUVs and LUVs are prepared in the same solution. So there's no change in osmolarity during the process, regardless of pores formed in the membrane. I hope to have answered the question. Does membrane proteins, like crowding proteins, not just pores, affect the dynamics of fusing and all the dynamics mechanisms you investigate? In our experiments, fusion is driven by membrane charges. So if we have anything that binds to either the GUVs or the LUVs, that will coat the membrane charges. So basically, we'll create a corona effect. Yes, we have investigated that. If we have proteins that very avidly bind to one or the other membrane, then fusion will be halted. So we don't have fusion anymore. And that's how it interferes with the fusion process. Not a question, just a comment. I admire your slides. Thank you. Very attractive visually, in addition to cool science. Thank you, Claudia. And these are all the questions for now.
Yeah, I think that's it. I just want to say you have really amazing microscopy. This was obviously a great scientific presentation, but I really appreciate the imaging, because that I can tell how much work went into it.
Thank you. Yes, it was a lot of work. I haven't done all the work. I've done most of the work, but I have had wonderful people also working with that in collaboration. I also performed the simulations. I did not perform any of the simulations, and they were done by Marco in Groningen, and he did an amazing job, and the videos they generate also outstanding. I love them. Another question popped up here. Should I continue?
Yeah, please.
What is the external versus internal solution osmolarity in your experiments? We know TXTL and pure have surprisingly high osmolarity. We always have to match with soluted outside. Do you do that? Do you do that too, or do you have hack around it? As I said, we use the same solution to prepare both the GUVs and the LUVs. That makes our situation much easier. If this is not possible in your experiments, if you have a solution that is of high osmolarity compared to the GUV, then you end up deflating the GUVs, and the fusion process deflates the GUV. So you have confounding factors. So ideally, what I believe is changes in osmolarity will not affect your reaction, but you will change the mechanical aspect. So you'll deflate the vesicles, you have membrane fluctuations just because you're deflating the GUV. Great presentation, thank you. Is it possible for the buds to bud out with the scramblazes during GUV growth? It is possible, depending on the preparation method. One always assumes that the GUVs formed are symmetrical. However, depending on the preparation method, you may also induce chemical differences. And these differences could be because you have certain lipid species preferring the upper or the bottom leaflet, depending on the preparation method. However, by default, we assuming that there's no bud information because the GUVs during their production are symmetrical. Oh yeah. What is the extent? I guess this has been asked.
Yeah, and someone asked, is it possible to get your PowerPoint? And I replied, but I realized I replied in the wrong thread, that we don't usually share the PowerPoint.
Yeah, that's correct, I think. There was another question that came later. Does the lipid specificity of the scramblase affect the GUV behavior and the calculated flipping rate? That's a very good point. We haven't tackled that. As I said, fusion and growth, in our case, is charged driven. We have worked only with the same membrane charges, so cationic LUVs and anionic GUVs, and we haven't played much with the GUV composition. I would not expect that to play much of a role because a lipid scramblase is fundamentally a lipid channel. So it's basically, it's not a hole in the membrane, but it creates a passage for lipids. And this is presumably very non-lipid specific. So I don't believe lipid specificity will play a major role unless you're working with extremely bulky lipids, such as glycolipids, and they will not fit the gap that you have in the protein. But this remains to be tested.
Awesome. I think I'm looking to see if there's any other questions. I think you answered all of it. And you also answered the question that I'm always wondering about, is how do you control osmolarity of those things? Because we're struggling with it all the time.
We had it easy. We had it easy for us. But I completely appreciate that that's not always the case. Also, there were many discussions in the context of the different synthetic cell consortia, which is putting all the different lines together. So in my specific case of growth, people want to couple that with division, and people want to couple that with metabolism and many other aspects. And one condition that works very efficiently for one package does not work very efficiently for the other package. So yeah, what I can say is that our system is very robust. But if we have a corona effect, a protein, for example, that binds membrane charges, then we already kill it. So we can work with various osmolarities as long as we don't have proteins, which is a very limiting, I understand it's a very limiting experimental condition for many.
Yeah. All right. If there are no more questions, I just want to say thanks so much again for your talk. And thanks everyone for these discussions. And have a great rest of your day, evening, whatever time zones you are.
Thank you, Kate.
Bye.
Bye-bye.